and sweaty.
It's gonna be great.
It'll be
We're
fun.
also excited.
Yeah, it'll be great because this is like my favorite topic. So,
Nice. It's 5:30 now. Do you want me to start with the intro or do you want to go it a little longer?
um, up to you. I'm fine with whichever. We can start now. And Laura will be the audience, which is fine.
All right. I'll start off with our land acknowledgement. Although land acknowledgement is not nearly sufficient in addressing these issues in their history, we would like to acknowledge that the Madison Mycological Society was founded on and operates within the traditional and current homelands of the Ho-Chunk people. Though the U.S. government attempted to remove the Ho-Chunk through coercive treaties and violent relocation campaigns, the nation persisted in fighting for their right to live with and protect their ancestral lands. We recognize and strive to combat the continuing white supremacy and colonialism in the fields of ecology, botany, and mycology, as well as the overwhelming culture of privilege and exclusion in outdoor recreation and education. And now let me introduce our speaker, Savannah Gentry. Savannah graduated from Oregon State University, where she majored in zoology before she went on to complete her PhD at UW-Madison, where she worked as a graduate researcher studying fungal pathogens of wildlife and focusing on host specificity and fungal physiology. She currently works as a research administrator for UW-Madison's Department of Genetics, as well as serving on the board of directors of the Madison Mycological Society. Today, she is presenting her work on snake fungal disease and an update on its impact as an emerging infectious disease. If you have questions during the presentation, please feel free to share or go ahead and share them in the chat on Zoom. And if you have any questions at the end, we'll have time for those as well. Without further ado, Savannah Gentry.
Okay, let me go ahead and pull it up. And I will say I cannot see anything when this is pulled up, so you'll have to verbalize with me. But oh, right. First, I got to share my screen, which is silly because we literally just practiced this. Let me share first. Screen, share, and then open and present. Okay. Can you, okay. Perfect. Okay. Yay. All right. Thank you, Ben, for the introduction. As he mentioned, I'm Savannah Gentry, and today I will be talking to you about snake fungal disease. And I really appreciate y'all letting me yap about one of my favorite topics. And also just to reiterate that questions are perfectly welcome whenever you have them. Feel free to interrupt me. Ben will also help in case you just want to focus on the chat and use the chat function. So I know that there is a question for me. Okay, so jumping in. So this is just a brief overview of kind of like the big pictures that I'll be talking about tonight. Things of like, what is snake fungal disease, the origin of the fungus itself, what we know about snake fungal disease today, and where do we go from here with this information. And there's a few other slides intermingled between, but these are the big points. So first, I want to start with a little bit of housekeeping. And then this is just a reminder of like some terms, if you're unfamiliar with them, just to give you kind of a little bit more context for my presentation. So starting with taxonomy and taxonomic classification. So if you're unfamiliar, taxonomy is just a way that we categorize and I use for identification of fungal species, and not just fungi, but of animals, bacteria, insects, and so forth. And so the biggest takeaway from this is that you start with the fungal kingdom, which is the most broad, and then with each different classification, it starts getting more and more specific, all the way down to individual species. Now, I won't be talking about each one of these. If anything, I'll just be focusing on order, and apart from talking about individual species. Additionally, you'll see some acronyms pop up like SFD, which just stands for snake fungal disease, and that's what I'll be referring to it as from now on. There's also EID, which you might see a couple times. That stands for emerging infectious disease, and I will define that later. And then some other terms that you might, it might look familiar, include hyphae. Again, hyphae are just the filamentous part of the fungus. It's what makes up its mycelium. And then there's conidia, which is just the asexual spores of fungi. And then more specifically, there's arthroconidia, which is just a type of asexual spore. And this is a spore type that's often used by pathogens. So I'll talk more about that as well. And then this is a term that you'll only see once later in my slides, histopathology. And that just means the study of diseased tissue in cells. So this is what a lot of pathologists use to kind of understand how fungal pathogens infect an animal or a person or a plant even. Okay, so starting off, I want to just kind of yap a little bit about why I care about SFD and why you should too, because it's really cool. But the first and foremost is that this is a global issue, it's a global disease. SFD is something that has become quite rampant in North America, especially in the Midwest area, unfortunately. But we also can find cases of it in many other places across Europe, Asia, parts of Australia, and so on. And so it's really becoming a concern for other mycologists like myself, who this is what our work was in, as well as conservationists who are trying to get in front of preserving some species that are really susceptible to it. Other reasons why this is important is because SFD is a really good example of a fungal pathogen that kind of is giving us an environmental cue of climate change. And so, as we know, climate change is warming the planet, and this is really causing a lot of reemergence or even new emergence of really severe disease that are caused by fungal pathogens. And so, what we're seeing is that there is a change happening with fungal physiology and pathogens like the one responsible for SFD, because generally we see it, and this is just an example, but we see this happening a lot with like temperature range tolerance. So, for example, the human body, our body temperature, our internal temperature is really high. This works really well for us as a first line of defense against fungal infections. So, whenever we inhale spores naturally, there's very little concern for us to contract any kind of fungal infection. However, with this kind of changing physiology and seeing that there are more pathogens that are starting to tolerate wider temperature ranges, especially on the hot side of that range, this becomes a big concern for medicine in terms of there's not really that many antifungals out there. So, especially in the medical fields, diseases like SFD and how it can cue us to be concerned about other pathogens is becoming more of an issue. Additionally, another reason why I care about this is what I spent my entire doctorate on in terms of the fungal physiology and trying to understand the animals that it infects. So this is near and dear to my heart. I will give this warning. I can't proceed without it. I will be showing you some images of snakes that have varying clinical signs of infection. And when I say clinical signs, it's synonymous with symptoms, but that's just kind of like a semantic thing in the world of research because humans are humans and they have symptoms, and everything else is called a clinical sign. We can talk about that later. But anyway, this is just to warn you. I will be showing some images. They're not going to be the whole presentation, but at least with the next few slides. Okay, so what is SFD? First and foremost, it's a skin infection that's caused by the fungal pathogen Ophidiomyces ophioidicula. And as you can see, it is quite aptly named because the direct translation of that Latin is the snake-eating fungus. And when we think of skin infections, when there's a fungal pathogen involved, they are often referred to as dermatophytes because it's of the skin. And so maybe that sounds familiar of like dermatitis. That's often caused by a fungal infection. Now, Ophidiomyces can be a little bit more than just a skin infection because it can persist for a while, especially in environments. But then also, depending on the susceptibility of the snake, it can in fact colonize further in its body, really disrupting internal organ functioning. So this can be a pretty nasty disease. And so, excuse me. And so, apart from that, Ophidiomyces is just a naturally occurring microbe in the soil, which is really interesting. And in fact, when there have been a couple studies done where it's kind of been put against other common soil microbes, and we find that it competes really poorly in the soil by itself. Now, there is the exception of when you're in a snake around a snake hibernacula. So if you find a snake den, oftentimes that is the case where Ophidiomyces is the best competitor. And that's because it's really, really good at degrading keratin. And so if that sounds unfamiliar, keratin is just what like we have in our fingernails, it's in our hair. It's also all over and inside a snake's body to different variations. So Ophidiomyces is really, really good at degrading keratin, which is one of the reasons why it can become such a nasty disease for these type of animals. And so I am going to just show you, this is a corn snake. And the reason I'm showing you this is that this is a completely healthy corn snake. This becomes important as I'm talking about SFD in general. But as you can see, yeah, completely healthy, nothing wrong with it. Now, these are pictures of a corn snake that's infected with SFD, and specifically in the areas that I have circled. And let me use my cursor. I think, can y'all see my cursor? Okay, perfect. So SFD does follow very similar disease progressions as any other skin infection might, in terms of there's a lot of swelling that's going to happen within the scales of the area that it is infected. And so it goes from swelling, then there's lesion formation that's happening. So if you can see, hopefully notice that there's the discoloration happening on the snout, as well as over here in this area. The whiteness of that is a lesion forming. And on top of which, when you have lesions form like this, there's a lot of bleeding happening, either just underneath the scale, or sometimes it can be so severe that it just creates a massive open wound. And so what is causing this is essentially Ophidiomyces. Once it gets an area of infection, it will start to cause cell death in that area and use that cell death to propagate itself more. And this cycle just repeats and repeats until that area of an infection becomes either really deep or much wider. So you can think of it as almost like a negative feedback loop. And so this is what SFD can present as in terms of like what you might see on a corn snake. But now we're going to move to a histopathology lesson. So again, this is the only time you'll see this word, histopathology, and primarily because I really want to show you what's happening on the inside when a snake is infected with SFD. And so what we're looking at is a really nice cross section of a healthy piece of skin from a snake. And so I'll walk you through this key. So see, there's my cursor. Okay, so this arrow right here is really pointing to this really thin line. Hopefully y'all can see what I'm pointing at. But that line, that's the epidermal layer. So that is the outermost layer. That's our skin. That's our first line of defense against really most infection or skin infections. And so where am I? And so this little asterisk, this is just the dermal layer. So it's just the dermis. It's right underneath the epidermis. And then this diamond is just showing you that this is the musculature layer. And the takeaways for this is that when we're looking at the inside, and especially as skin samples from snakes, a healthy epidermis is going to be nice and thin. This is important that you remember. And so the epidermal is thin, and we see a beautiful differentiation of all of the skin layers. So that's what makes a healthy skin sample. And the reason also that it's just pink is because this is dyed. So there's different dyes that are used in histopathology to help both differentiate skin layers, but also just to show us what's happening when something is happening at all. Okay, so remember, this is a healthy piece of skin. And this is what happens when a snake gets infected. So I'm going to start with the image, which with image C. See, what is it? There we go. And so hopefully you can recognize that it does look much different than the healthy piece of skin. And so this arrow right here is pointing, don't mind me, pointing to the dermis. Now remember, a thin dermis or a thin epidermis, my mistake, is healthy. Here we see that the epidermal layer has become extremely swollen, and that's inflammation that's happening. So that's what we're seeing in this image. We see a lot of inflammation in this area, and there's even some inflammation over here. Uh, and so the arrowheads that you see in image C, this is just showing you that there is an immune response happening. So all of the white blood cells are activated, and how we know that is, if cursor, this kind of purpley blue color, that just means that, yeah, the white blood cells are not happy and they're working overtime to try to get rid of the infection. And so in image D, that's just a close up of this area. Where this dark, dark magenta is, this is what lesions look like on the inside. So recall that, that white kind of color when a snake gets a lesion on its skin, this is what's happening right underneath it. And so what we're looking at is that these lesion areas are filled with arthroconidia. And so you can see that it's just propagating, and more so with these arrows right here. Hopefully you can see the dark pinker lines. I really hope they show up well for y'all. But these kind of dark pink lines, those are hyphae. So we have hyphal fragments that are growing from the arthroconidia that just get split up as best as they can to get further and further into the skin layer. And so that's one of the reasons that pathogens that have arthroconidia, like Ophidiomyces, are so successful, because you just need one for it to start growing. And so this is what's happening on the inside of SFD. Okay, so now that I've shown you kind of what it looks like externally and internally, we're going to play a little game. And this is called Can You Find SFD? So I want you to look at these two pictures. These are two pictures of of water snakes, and these are snakes that I caught on a surveying trip. So excellent pictures, thank you. And so take a few seconds to look at these pictures and pick which one you think is positive for SFD. And there's only one. There's only one that's positive. So I'll give you a few seconds while I take some sips of water. Okay, so if you guessed that the bottom picture is a snake that is positive with SFD, you would be correct. And most precisely in this area. So if you can see, it's right within this red circle that there is basically an open wound. There is some necrosis happening, so cell death happening. It's a little weepy. And this is often what we might see in wild snakes that have SFD. And again, and I'll speak more to the variation of clinical signs, but this is very common that we might see. Now, if you pick the top picture, it would be wrong, but it's still a good try. This is a snake that it was not positive for SFD. And if you thought that maybe it was this area, that is actually just a scarred bite mark. And so the whole point of this game is really that I hope it emphasizes how difficult SFD can be to recognize in nature. And this is something, even when you know what you're looking for and how it might present, can be very difficult. And so when you have research happening or kind of surveys like this going on, we often couple our observations of any clinical signs we might see with sampling. And so this is just to help us have more powerful and accurate understanding of when we're seeing SFD and not, because the big caveat of this is that you can have snakes that don't overtly show a clinical sign of infection, but still be positive for SFD. And I will talk more about that in a little bit. Okay, so I also wanted to mention that an important fact about Ophidiomyces is that this fungal pathogen is considered a primary pathogen. And all that means, what makes a pathogen a primary pathogen is because you can be completely healthy and get infected by this fungus. Now, this is makes it particularly, in my opinion, very cool because oftentimes, and I'll use, us as humans as examples, oftentimes we don't get fungal infections unless we're already sick. This is, if there's something else going on, if we're immunocompromised, this is why immunocompromised people might die from a fungal infection. Ophidiomyces doesn't need any of that. It'd be like, I think I'll infect. You can just have a thought and then start to infect you, no matter how healthy a snake in this case may be. So the next part about SFD is that you can have snakes recover from the infection. Now, even though there is recovery, there is a high chance of disfigurement in the areas of infection. So here I'm showing you another picture of a corn snake. This is a corn snake that has cleared infection. And in the little red circle, you'll see that there's just some slight discoloration and the scales are a little warped on the tip of the snout. This is often what will happen, no matter how severe or unsevere the clinical signs might be, there's always going to be some sense of disfigurement in the areas of infection. And I will say, prepare yourselves because now I'm going to show you a slew of images. This is an image of different snakes. These are all different species, and I hope it's not lost on you the variation that I've been talking about. And so you can see that looking at image C right here, this is a rattlesnake. So I think this one is a Timberlake rattlesnake. And so they're very susceptible to SFD and probably suffer most severely in terms of the species that are infected or that can be infected. And then as you can see in comparison, this snake has just a few lesions. It can maybe shed them when they shed its skin. But yeah, the whole point of this is to really emphasize like there's a lot of variation in clinical signs. Sometimes snakes can survive it and sometimes they can't. And even more on this is that naturally a snake like some lizards will naturally shed their skin. These animals will also shed their skin if they come down with any kind of infection, whether it's fungal, bacterial, or viral. Essentially, they get cued. And so the whole point of this is that they shed their skin as a way to hopefully clear whatever is causing the infection. Ophidiomyces is so persistent that even if a snake sheds, it will remain. And that's what causes a lot of the reinfection that we might see in some species. And so it just makes it that much harder for snakes to recover. And there's no guarantee of if a snake recovers once, it will survive the next time it's reinfected. And this next bullet point hints at that there is no snake that is immune. And essentially, there's any population or any snake species that's been recorded with SFD, they're all different. It all can be from these places around the world. So it's really can be a really difficult disease to even treat. And when I say treat, there is no treatment. So oftentimes we're just stuck with this question of how can we make these animals as comfortable as we can make them in the hope that they can clear the infection on their own. And so this is just another picture. I'm again to emphasize how difficult it can be to see SFD in nature. And this little bump is where the area that was positive for SFD. So it can be difficult to survey. That's why we kind of couple these techniques together between sampling and sequencing and just clinical observations of these animals. So hopefully this acronym looks familiar, EID. SFD is considered an emerging infectious disease. And what that means, or is defined as, is that it can be a newly appeared disease or a pre-existing disease that we already know about, rapidly increasing in geographic range, susceptibility, and incidence. And unfortunately, with SFD, it fulfills all three of those categories. And you don't need to have all three to be considered an EID, but SFD is that amazing, apparently. So it's an EID in the same way that chytridiomycosis is an EID. And hopefully, if that doesn't sound familiar, that's okay. But chytridiomycosis it's been in nature, it's been on the news over the last few years. This is a big issue because this is a disease that has impacted huge amounts of biodiversity in North America, especially in amphibians and salamanders. And well, salamanders are amphibians, I meant frogs and salamanders, but all amphibians in general. And so this is caused by a fungal pathogen. It's caused by Batrachochytrium dendropatitis. We just refer to it as BD because that's easier. And I think the last time I recall like seeing any statistics about chytridiomycosis is that at least to date, it has caused 90 different species to go extinct and up to 500, if not more, to be on the brink of extinction. So this is a really big problem. Additionally, SFD is a EID just like white nose syndrome. And again, if you haven't heard about that, that's perfectly fine. White nose syndrome was a really big problem for our bat populations. I would say since the statistics of like 2023, to date, it's caused 6.7 million deaths in our North American bat colonies. And that's huge. That's a huge biodiversity loss. And the fact that we have the same problem with SFD is becoming a big concern in terms of helping conserve, especially some of the more susceptible species. Okay, so now we're going to move on to the origin of Ophidiomyces. I really love talking about the origin, not particularly because it's a unique story. As I'm talking about it, this is a story that a lot of fungal pathogens these days follow. And as you can see with this meme on this slide, that taxonomy is a beast. It is constantly changing. I think those of us who do any kind of foraging or identification can appreciate that. And so Ophidiomyces is no different. So our story begins with the fungal complex that was once called the Chrysosporium anamorph of Nanesiopsis reesii. That's very long. All you have to remember is that it's called the CAN-V complex. That's what we refer to it as. And so what a complex like this is and why they were used a lot, especially before we really had the technology to help identify species from any samples, is that in the case of CAN-V, there was a fungus, a fungal pathogen called Nanesiopsis reesii. And ever since that fungal pathogen was found, any other pathogen or any other fungus or sequence that maybe cause similar issues or look somewhat morphologically the same, but we're not sure, they all get lumped into a complex. And then like, we'll deal with it later. So that's essentially how fungal complexes worked back then, especially before the whole advent of sequencing samples. And so the CAN-V complex sat in the Onigenales order, harkening back to taxonomic classification. And the Onigenales order is my favorite fungal order because maybe unsurprisingly, this is where most of known fungal pathogens reside. And that includes pathogens that infect us, animals, all kinds of animals, and other fungi, and even some plants. And so CAN-V essentially housed any of these fungi that infected snakes, lizards, turtles, and more, including ones that infected people, which seems strange. And so really, like I said, like we really had to wait for technology to catch up for any species identification to kind of be parsed out of this complex. And Ophidiomyces, before it was named Ophidiomyces, any report or article that was being published about something similar, they were seeing snake populations die suddenly. They didn't really know what it was. So those type of reports we really start seeing in the 1980s. And so that's a long time for us to not know what's going on and really allow Ophidiomyces to kind of gain traction as an EID. And so uh, as I alluded to, like before Ophidiomyces was Ophidiomyces, it was the Canby complex and then got parsed out as like a Chrysosporium species. We don't know what, but we know it's different than the Nesiopsis reesii. And then I would say in 2013, if not between 2006 and 2013, is when Ophidiomyces got its name. That's a long time for that to happen. So we're still kind of on the recent side of it being Ophidiomyces. And so this might lead to a question of like, no, okay, well, if it's been here since the 1980s, was it always an EID? And the answer is that it's very hard to know. It's a very hard question. It kind of remains like this open-ended question, and that's because of two functions. So when we hear about an EID and it starts to become more known, we start to go out and look for it. And generally, the more you look, the more you will find. So even though it's been here for a while, this is a trend we often find with pathogens like these. Oh, we know what it looks like now. Let's go out and find it and see if it's in the environment, and it usually is. And that makes it, especially in the case of SFD, really hard to determine if it was like an introduced disease, like for example, white nose syndrome. So white nose syndrome in bats is caused by a fungus that is really contained to cave systems. It's not something that's easily found in the environment. But because of the mass loss and not extinction, but mass die-offs that people were seeing, really quickly we were able to determine that white nose syndrome was an introduced pathogen. And funny enough, and maybe unsurprisingly, it was introduced by people. And because it was introduced by cave splunkers, people who enjoyed going into caves, the spores would just stay on their clothes, and by the time they go to another cave, they're taking the pathogen with them, and that's how white nose syndrome was introduced. Unfortunately, SFD is not so clear cut. I wish it was, but it's one of the reasons why SFD is such a difficult disease to even contain, let alone treat. So, what do we know today about SFD, especially now with the power to identify? Uh, because we can sequence the samples that we, being like researchers or any collectors, this has really given us the power to trace where we can find SFD. Snakes in general, especially in the wild, are difficult to survey. Most of the time, they're solitary creatures. You know, you can't really find one unless you're circling the same area 5 million different times throughout the year. So this has really been a big proponent of how we know that SFD is here and has been here. And so this also helps us understand seasonality. So one of the similarities between SFD or between Ophidiomyces and what causes white nose syndrome in bats is that both of these pathogens really take advantage of when the animals are going into hibernation, or in the case of snakes, brumation. So brumation is very similar to hibernation. The only difference is that snakes still will wake up to go feed themselves and then come back and, slow all of their systems. And so this is a really perfect time for Ophidiomyces. Recall that it gets really concentrated in the dens that they create for themselves. And so when they're not moving a lot, when they're sleeping a lot, Ophidiomyces will colonize their skin pretty severely. And so because of that, when snakes are coming out of brumation, it becomes easy for us as researchers and surveyors to have to kind of plan our surveying around their brumation times. So we can get an idea of the concentration of what's happening in these populations and trying to see how often snakes are getting reinfected. The other thing we know is that SFD is a huge problem within the exotic pet trade. This is, again, one of the reasons why it can be so difficult to track in terms of where it's coming from and where it's been. And that's because both of like collectors, you have people who own snakes like myself, and then also zoos are having a very hard time right now, even helping their own snakes kind of survive SFD because of all the trading that happens among zoos also. And one of the things, though, that we can say, and it's been really, really cool, there's been a few papers that have come out that have used sequencing and sampling of old museum specimens to sample if they're positive for Ophidiomyces. And we now know that it's been here since the 40s. So it's been here even longer than we initially thought, which has a lot of implication for the ecosystems that it's been living in. And so, how this all kind of occurs with like fungal physiology. We also know that SFD or Ophidiomyces no longer just affects snake. This is what all my work was a part of in terms of figuring out other wildlife or animals that it infects. And so now we know that Ophidiomyces causes infection in bearded dragons also. And why this is important is because bearded dragons are a very big part of the exotic pet trade. They make up a very large portion. And so you have the trade and transport of these animals. There's not a lot of regulation or easy regulation that can be applied to the exotic pet trade. So it just makes it difficult for us. And so one of the things that this is helps us understand is that okay, so bearded dragons can get infected. So what are the other potential hosts that could be infected? And you'll notice I have reptiles, I also have birds on here, and that's because of the origin of Ophidiomyces being from that Canby complex. Ever since then, it's been parsed out into numerous different pathogens that affect different reptiles. And so if Ophidiomyces can infect other hosts, there is a high probability that some of these other pathogens can do this cross-contamination like this. And that has a huge implication on ecosystems and how this disease is transmitted and remaining so persistent in the soil. And on here, I also have like, is it a reservoir host? And what that means is just that it could be an animal that is transporting this fungus without getting any infection. This is kind of a common way that there might be pathogenicity being spread across different environments is because there's some unknown host that might be spreading it but not getting infected. So that's also a question that we try to understand with SFD. And so, like I said, there is no treatment. Really, there's no good treatment to SFD. I think there's maybe one paper that might have looked at treating it, but with little success. So unfortunately, this is something that we really hope the animals just have to clear on their own. And one of the things, I don't think I mentioned this, is that SFD, because it infects wild snakes and captive snakes, the chances of a wild snake surviving is far less because with captive snakes, like my pet snake, I feed her regularly. She wants for nothing. So she really has the best chance of clearing this infection without any intervention by me. And so the other thing we know now about SFD is that North America has a different strain of Ophidiomyces than Europe. And how it affects our snakes in all of North America is far more severe than how it infects snakes in some of these other countries. And this is also a similar trend of what happened with white nose syndrome. When it got introduced over here, none of our bats were accustomed to the pathogen, whereas in Europe, those bats had evolved with the fungus. And so it makes it a lot harder. They're way more tolerant of the infection. And so that's one thing we know now, especially with the sequencing. I know I've said that a lot, but the severity in North America is much different than some strains in Europe and in Asia. All right, so where do we go from here after all of this information? So I think the biggest question and biggest way to keep in mind how we can be better about SFD or make sure we're not spreading it in the way that we have done with white nose syndrome is just learning how to be good stewards of the ecosystems we like to visit. And so there's also a big one through research. That's a great way. One of the, I would say that sequencing can kind of be a double-edged sword. It's a great, powerful technique that helps us know more. But because of that, there's also less and less physiological experiments happening with fungi in general. And that is such a good way and a good basis for us to get as much information about a fungus as possible. So knowing what it grows on, what's its morphology look like under different conditions. So there's definitely a big need for more of those type of experiments. Also, because there's no treatment, trying to understand infection recovery even more in terms of how we can give animals the best chance, maybe in the from the wild versus what we're doing in captivity, and identifying other potential hosts, like what I did with bearded dragons, trying to see, okay, is there other ones? And I say this because so when I showed you the SFD game, those two water snakes, in that same area in the environment, those water snakes were living among green anoles. And any kind of sign of clinical disease that was ever seen in those green anoles is all word of mouth. No one has actually taken the time to study that and sequence if there is an interaction happening between these two animals that causes even more dissemination of infection. So again, much need for more research on SFD. So how we can do this in terms of volunteering is a few ways. One, and this is a way that I've worked with the National Wildlife Health Center that's here in Madison by collecting and submitting wild snake skins. So anything that you find in nature, granted, of course, making sure that it's not completely destroyed, but this is something we can do. We submit them and they'll get to it at some point. USGS research depends on what the government is doing. So keep that in mind. Also to report where you're seeing snakes in nature. So if you're going on a hike, if we're going on a mushroom foray and you happen to see a snake, logging a report is a good way because that again helps us understand, okay, so if this species of snake is in here, what's the likelihood we can try to find Ophidiomyces and if not an infected snake. Also by having good steward habits. And what I mean by this is that we want to avoid a white nose syndrome situation. Once you leave an ecosystem, a really good habit to have in the case of SFD is just making sure you desanitize your hiking boots or whatever shoes you're wearing. And now to that end, all you need is like a 10% concentration of bleach, and that will do the job. And so being aware of those type of techniques, especially as you're going to different ecosystems and environments and just enjoying recreation, it's important to keep that in mind. Also, combating bad perceptions. There's a lot of people who hate snakes and they think they're slimy, they think they'll just bite them, and that's never really the case. Snakes are extremely important keystone species in the environments that they live. And a keystone species is just that once that keystone species is gone, the ecosystem degrades. They're very good at keeping rat populations and other vector populations down. And they're very important that we maintain their species as a whole, all different snake species. And so we do this through awareness, like me giving this talk, trying to say like, oh no, you don't need to kill that snake. Leave it alone. It'll leave you alone. And then just also recognizing snakes can be cute. They're just cute little boop noodles from time to time. I certainly think so. But so with that, I will say thank you so much for listening. This is just a quick thank you to show all of the people and organizations that I worked with to do my own work. and I really hope you enjoyed this talk. So I will take any questions now. Let's see, and I'll stop sharing. Okay. Whew, I did it.
I have a question or two if no one else does.
Yeah, go ahead.
Do we know if snake fungal disease prefers living versus dead hosts? Does it
Mm.
spread more on dead ones?
I think so. It's not so much that it prefers, it is easier to eat a dead host than fight its immune system, is probably the better way to think about that. So it's like it'll still try to colonize something that's alive, but it'll have a much more successful time eating the keratin or whatever of anything that's already dead.
Ah, okay. Interesting.
Yeah, so it's definitely one of the reasons why it is also considered like a good degrader in the environment because it can degrade keratin, which is a very strong compound in general. It can be difficult to degrade. Let's see. Yes. Joseph, I think your hand is raised.
Oh, yeah, I had a question. Is there like the histopathology or what kills the snake? Is there like secondary bacterial infection that the snake dies from, or is it like blood loss, or do you know?
Right. That's a great question. So I will say, especially in wild snakes, what is often the case is that there can always be maybe like a dual infection happening in a wild snake, but what ends up being its demise most of the time is that SFD and like the fungus itself really likes to colonize the face and the snout area. So it'll build all of these painful lesions around the mouth that will prevent it from eating. And, from the rattlesnake picture, sometimes they'll even go blind, and it just makes it really, really hard for them to hunt. So when they do get infected, often what causes them to die is just starvation.
Thank you.
Yeah. Thanks for the question. Andrew, did you have a question?
Yes, I have multiple. I think the first one that I'm most curious about. So like you shared that there isn't really a treatment, which is sad because the whole time you were talking, I was like, couldn't we like give them a bath like with like I don't know, like some Selsun blue or something to like kick out the
Hmm.
fungus? But I don't know. I guess that's not really a thing. So one question that I had is also just like if you were trying to culture this fungus, like what kind of media does it grow on? I know like
Hmm.
so it grows on it can apparently still colonize like dead snakes, but
Yeah.
like how do you just
It
to
can grow
study
on
the
a
thing, I'm like, yeah.
Yeah, it can grow on a lot of stuff. Like because it's a soil microbe, even though like
Mm,
keratin is well, that's its most yummiest treat and it loves it the most. It can still be grown on like oh my God, it's been years. Potato dextrose, like the very
mhm.
standard stuff that's used in the lab, as well as like really specialized media. So it doesn't need keratin to grow. It just does really well with keratin.
That makes sense.
Yeah.
Yeah, and it's interesting too. I mean, I guess like that kind of clears up another question I had too, which was just around like, so this thing is in like the water, like it's everywhere, right? Like it's
It's
getting water
everywhere.
snakes, it's
What?
getting yeah.
Excuse me. Yeah, and so I know there's like some questions right now of if it has some kind of adhesive properties that other fungal pathogens might have, especially ones that infect people, uh, as to why it can persist so well on in different environments and even after a snake sheds its skin. Anything else?
That's important.
That's
Oh,
all I have.
I have one more question here. Yes, what is it?
Um,
I have two friends here too who are listening.
from the genetic sequencing, do they have an idea of the origin based on like the genetic diversity of
Mm.
fungus?
Yes, that's a great question. In case you did everyone hear that or no? Okay, cool. Yeah, so even though we have different strains, there's been a paper, at least within the last 4 years, that came out that really kind of shows where its evolutionary path is from a strain in Korea.
Oh.
So it's not, that doesn't mean that it was introduced from Korea like recently, but it does have a lot of similarity to a strain that is in Korea. And I remember when that came out, everyone was so thrilled, just like, oh my God. Yeah. So
So they think it originated from Korea?
I don't know if that's like the hardest question. It's I think it's like the closest relative. So whenever it came over to North America, wherever that might have happened, it at least its oldest relative is in Korea.
Okay.
So
Cool.
yeah. Anything else?
I've got one other. Oh, sorry. Go ahead.
Oh, you can go first.
Okay. You mentioned that it's an anamorph. Do we know the teleomorph stage of it, or is it it's just
No,
anamorph that
we
we
do
know?
not. And
Oh,
that's
okay.
one of the
Interesting.
reasons why we might think it's so like our snakes, apart from the evolutionary detail of the snakes in North America not really evolving with this pathogen, is that we don't know what its reproductive state looks like or like the sexual reproductive state. And we feel like if we know that, then we can ask better questions about like why is the asexual state so severe for our snakes? So we don't know that yet, but I think we're getting closer,
Oh, very cool.
hopefully.
My question was, what sort of impact is this having on the snakes in the environment? Are there certain areas where, it's decimating the population? And is it specific for the corn snakes, or does it go to, other snakes?
Yeah, so this is a disease that's not specific to corn snakes. It impacts all snakes. There is no snake that's immune. There are different levels of, variation in maybe how resistant a species is. But I would say for sure we're seeing a lot of decimation in the Midwest. That's where a lot is, at least in North America, particularly with rattlesnake species. And if the Eastern Massasauga, that's a species that has a lot of concern right now. And like timber rattlesnakes, their populations are in mad decline because of this disease. So there's a lot of like joint effort between some of the research happening and some conservationists trying to figure out how do we save these snakes? What do we do? So yeah, there's definitely some species it impacts far more than others. And although we don't right now there's a lot of research going into kind of the natural defenses a snake has. We don't have a really hard answer on why rattlesnakes are so susceptible right now.
And does it affect, I mean, so does it affect like the prey that these snakes, is there an ecological upsurge of like small rodents that like these snakes that feed on, that they don't have that pyramid anymore, so and also, are rodents infected with this and are they dying from this also?
That's a great question. And unfortunately, we don't know the answer. No one has looked. I think I was the first person to look at SFD at all infecting another reptile. There's been no work done on if there's any transmission between the snake and what it eats. Or like if it gets involved with the rodent and the rodent survives, is the rodent infected? So those are all questions that remain unanswered. I would say right now there is probably some, I say contamination, but dissemination of disease happening between snakes and lizards in the area for sure. But we also see that when a snake lays her eggs, for example, it's not direct vertical transmission like through the fluid, but if a snake has SFD and then she lays her eggs, that Ophidiomyces will be all over those eggs
Hmm.
when they hatch. So, yeah, there's a lot of unanswered questions.
And what about in the rest of the world? Like, you said like in Korea or like other parts of Asia, is this wreaking havoc amongst those wild snake populations? And then has it been seen in like the zoos, where snakes are kind of like, oh,
Yes.
and
Ah, yes, I'm loving all of the questions. Yes. So yes and no in the sense that European snake populations in the wild seem to be handling SFD a lot better than the snakes we have here in North America. And I think as I mentioned before, this is just a theory. We don't have evidence right now, but how evolution happens with some of these diseases, if there is a host that tends to evolve with the pathogen, it can tolerate it a lot better than some of these species that have just never seen it before because it hasn't been an issue until now. And so in Europe, I say the wild snakes are doing much better, but we are seeing it become a huge problem in zoos all over. There's trade and transport of these animals. I would say every year there's a new paper that comes out, if not two, about, oh, we found it in Taiwan. Oh, we found it in Japan. Oh, we found it in Italy. And so there's more and more that keep coming every time. And most of the time, those types of studies are coming from case studies from veterinarians, so like people who have pets in zoos. Does
Thank
that
you.
answer your question?
Yes, thank you.
Okay. All
I have one more question.
yes, one more
Okay,
question.
and I
Yes.
know this might be based on like data collection, but have they noticed higher instances of infection with climate change? And again, I know it could be biased based on sampling, but
Yeah, that's a good question. I think in the sense of like climate change, the biggest evidence that we're seeing is just that there's other diseases like SFD becoming an issue. And so it's hard to say since this has been around since the 1940s. But now with climate change, it's almost like at what point have we reached that maybe it wasn't a problem and now it's exploded into a huge problem? So I think there is definitely some climate influences there, but I think that's just all we can kind of guess at right now.
Oh, I
Okay.
had one. So has anyone looked at, the giant reptiles? And, I know the theory is, oh, it's because of the meteor and, all the clouds and stuff. Has anyone looked at the fossil records to see is there the DNA for this particular fungus in those fossils?
Yeah, kind of. So like actual fossils, no, but like old museum specimens, absolutely. And so that's how we know right now that Ophidiomyces has been around since the 1940s because we've had researchers sample old museum specimens from back then. And so that's like that was a amazing finding. But I wouldn't be surprised, like the more museums, if there were more samples, especially in other areas, we might see a similar thing. Just based on the fact that Ophidiomyces is in the soil naturally.
You should contact the Field Museum in Chicago because they have
Mm.
a whole DNA analysis lab. And I mean,
Yeah.
have you visited there?
I've never visited there.
Oh,
No. It
yeah.
is
So, I mean,
definitely on the list.
yeah, no, and I mean, there's this amazing, stories that they talk about what they've discovered, like in the DNA of, but not going back to the dinosaurs, So I think, and they have a lot of nice dinosaurs, you know,
Nice.
specimens, or artifacts, that it can easily be tested, and they would probably love to do something like that.
Yeah, that is awesome. I do know someone at the National Wildlife Health Center that they're the person who did like one of the museum papers. So I'll have to ping him to see him like, hey, did you get him from here? Yeah, I think we have time for one more question. If there's anything, we're almost at time. Anything? Okay. Well, if not, I really appreciate all the questions. This has been such a joy. Thank you so much for having me to talk about SFD. This is again my favorite topic.
Thank you. It was fascinating.
Good, I'm glad.
Yeah, thank you very much.
Yeah. Have a good day.
You too. Thanks so much, Savannah. That was so
You're
awesome. Oh my
welcome.
gosh.
Yeah, I'll close out the room and get the recording as well.
Sounds good.